Hello friends, welcome back to our new video, that is MCQs on biochemistry, and this is the part 6 of this series of videos on biochemistry. So the first question of this video is, which of the following diseases are associated with genetic or nutritional deficiencies that alter protein maturation process? And the options are mad cow disease, Alzheimer disease, bovine spongiform encephalopathy disease, or all of the above. So the right answer is option D, that is all of the above are the correct. So these all diseases are associated with genetic or nutritional deficiencies that alter protein maturation process. Okay, so let's move on to the next question, that is question number 101. So, which of the following statement is correct for a reaction A plus B reversible sign equals to AB? So, options are: larger the value of the equilibrium constant, weaker is the binding between A and B substrate. Okay. Option B: lower the value of the equilibrium constant, stronger is the binding between A and B. Or option C: larger the value of the equilibrium constant, stronger is the binding between A and B. And the last option, that is option D, this is a third order reaction. So the right answer is option C, that is larger the value of the equilibrium constant, stronger is the binding between A and B of this reaction going in A plus B reversible, that is AB. Okay, so let's move on to the next question, that is question number 102. So the amino acid with 5 bond and shy bond values, that is minus 60 and minus 40 and minus 59 and minus 47 and minus 80 and 120. So that will be adopting which of the following conformations? So 5 value and shy value of like this will be adopting which of the following conformation of secondary structure? And the options are helix helix extended form, or helix coil extended form, or extended loop form, or loop loop coil form. So the right answer is option A, that is helix helix extended form. And the reason behind this, because here we can see this is the Ramachandran plot. This is the Ramachandran plot, and in our question we can see that most of the phi value and psi value are in negative, psi negative and phi negative. So psi negative and phi negative quadrant is this one. This one is the phi negative and psi negative quadrant. So here right-handed helix or helix form are more preferable. So that's why we have to choose helix helix extended form. Okay, so that is the reason. So now let's move on to the next question, that is question number 103. So which of the following proteins primarily participate in folding kind of work? And options are beta helix, alpha helix, chaperone protein, or HSP, that is heat shock protein. So the right answer is option C, that is chaperone protein. So chaperone proteins primarily participate in folding kind of work. Let's move on to the next question, that is many polypeptides are initially synthesized as larger precursors called as option A prosthetic group, option B apolipoprotein, option C proproteins, option D complex proteins. And the right answer is option C, that is proproteins. So proproteins, so many peptides are initially synthesized as larger precursor called proproteins, and then they are cut into the main active enzyme. Okay, so that is the larger precursor is known as the proproteins. So let's move on to the next question, that is methotrexate, an analog of dihydrofolate, is an inhibitor of dihydrofolate reductase. So methotrexate would be expected that is option A, decrease the Vmax of the enzyme, or option B, increase the Vmax of the enzyme for the substrate, and option C, decrease both Km and Vmax of the enzyme, or option D, increase the affinity of the enzyme for the substrate. And the right one is option C, that is decrease both the Km and Vmax of the enzyme. So methotrexate, an analog of the dihydrofolate, is an inhibitor of dihydrofolate reductase, and it decreases both Km and Vmax of the enzyme and inhibit them. Okay, so let's move on to the next question, that is polyproline 2 structure found in some proteins and having helical orientation is known to have option A 3 residues per turn, 3.3 residues per turn, or option C 3.6 residues per turn, or option 4 residues per turn. And the right answer is option A, that is 3 residues per turn. So polyproline 2 structure found in some proteins and having helical orientation is known to have 3 residues per turn. So let's move on to the next question, that is if the pK of the epsilon amino group of lysine is 10.5, the side chain at pH 7 is likely to be option A neutral, option B half dissociated, option C negatively charged, option D positively charged. And the right answer is option D, that is positively charged. So if the pK of the epsilon amino group of lysine is 10.5, the side chain at pH 7 is likely to be positively charged. How? Here we can see. So this is the structure of lysine. Okay. So here here mention the epsilon amino group of side chain. So this is the main core structure of the lysine, and this is the side chain. So here side chain, then the epsilon position after 4 CH2 group, one NH3 group is present. Okay, and here we have said that they said that the pK value of epsilon amino group is 10.5. So when the pH is 7, then what we can see in the NH3 group will be positive sign or negative sign. So the so this NH3 group, NH3 plus group, release their H plus ion when the pH will be 10.53. Okay, but previously when the pH value is 7, then pH value when 8.95, then they release the second NH the H plus ion from its amino group. And firstly at pH 2.18 they release the H plus ion from this COOH group. First they release the hydrogen ion from COOH group, then in pH 8.95 they release the hydrogen ion from amino group from here, and then after 10.53 they release their hydrogen ion from the side chain amino group. So when the pH is 7, that time the amino group which is present in side chain will be present in positive form, because in that time they do not release the hydrogen ion. That's why they present at their positive form. So positive charge will be the right answer. Okay, so let's move on to the next question, that is 108. So the dihedral angle psi for a peptide used in the Ramachandran plot is given a value based on a rotation around N C alpha bond, option B C alpha C bond, option C C and N bond, or option D C alpha C beta, sorry, this is C alpha C, sorry, C alpha C beta bond. So the right answer is option B, that is C alpha C bond will be right answer. So dihedral angle psi for a peptide used in Ramachandran plot is given a value based on rotation around C alpha C bond. So how we can see here in the picture. So here you can see this NH, so this CO and NH is the peptide bond, and here the C alpha, this is the C alpha, sorry, this is the C alpha and C. So this bond is called the Chi bond. So this is the Chi bond, okay. So that's why it is called a Chi bond between the C alpha and C of the peptide bond, okay. So that's why this is the reason, that's why we can say the Chi bond is C alpha C bond, okay. So let's move on to the next question, that is 109. So which of the following consists of heme group? And options are myoglobin, hemoglobin, both A and B, neither A nor B. And the right answer is option C, that is both A and B is the correct one. So myoglobin and in hemoglobin, heme group are present. So let's move on to the next question, that is question number 110. So template theory of enzyme action is supported by option A, compounds similar to substrate inhibit enzyme activity, or option B, enzymes determine the direction of reaction, option C, enzymes speed up reaction, option D, enzymes occur in living beings and speed up certain reaction. So, and also here we can see that the template theory of enzyme action or template theory is also known as the lock and key. Also we can see lock and key theory, okay. So the right answer is option A, that is compounds similar to substrate inhibit enzyme activity. So in template theory of enzyme action is supported by compounds similar to substrate inhibit enzyme activity. So let's move on to the next question, that is a coenzyme is organic or inorganic group that is essential for enzyme activity, or option B, that is organic non-proteinaceous group that is essential for enzyme activity, or option C, one that share function of another enzyme, or option D, same enzyme found in different organs or tissues. And the right answer about the coenzyme is option C, that is coenzymes are one that shares function of another enzyme. So in different enzyme, same coenzyme can be act. So let's move on to the next question, that is the reciprocal coupling of proton and oxygen binding is referred to as option A, anoxic effect, option B, Bohr effect, option C, Hill's effect, option D, bond effect. And the right answer is option B, that is Bohr's effect. So the reciprocal coupling of proton and, or proton we can say pH, and oxygen binding is referred to as Bohr's effect. And here we can see the oxygen binding affinity of the hemoglobin is inversely related to acidity or pH and carbon dioxide concentration, okay. So this is the main concept. So let's move on to the next one, that is double hydrogen bond occur in DNA between option A, adenine and thymine, option B, thymine and uracil, option C, thymine and cytosine, option D, adenine and guanine. And the right one is option A, that is adenine and thymine. So in between adenine and thymine, two double bonds are present, and in between guanine and cytosine, three double bonds are present, okay. So let's move on to the next question, that is question number 114. So multiple molecular forms of enzymes occurring within a plant system are known as option A, vitamins, option B, coenzymes, option C, hormones, option D, isozymes. And the right answer is option D, that is isozymes. So multiple molecular forms of enzymes occurring within a plant system are known as isozymes. So isozymes, which are enzymes, their molecular structure are different but they perform the same reaction. Okay, so let's move on to the next question, that is question number 115. So which one of the following enzyme requires molybdenum? And the options are carboxylase, option B hexokinase, option C flavin oxidase, option D nitrate reductase, and the right one is option D, that is nitrate reductase. So nitrate reductase, which is an enzyme which requires molybdenum. Okay, so next question is which one of the following is a mitochondrial marker enzyme? And the options are aldolase, amylase, succinate dehydrogenase, pyruvate dehydrogenase, and the right one is option C, that is succinate dehydrogenase. So succinate dehydrogenase is a mitochondrial marker enzyme. Okay, so next question देखेंगे, which one of the following enzymes is used in PCR reaction? And the options are Taq polymerase, RNA polymerase, ribonuclease, endonuclease. And the right one is option A, that is Taq polymerase. Taq polymerase is used in PCR reaction. Next question here. In the study of enzymes, a sigmoidal plot of substrate concentration versus reaction velocity may indicate Michaelis-Menten kinetics, cooperative binding, competitive inhibition, or non-competitive inhibition. And the right answer is option B, that is cooperative binding. So in the study of enzymes, a sigmoidal plot of substrate concentration versus reaction velocity may indicate cooperative binding. So let's move on to the question number 119. So in the thylakoid membrane of cyanobacteria, algae and green plants, the thylakoid membrane contains two kind of chlorophyll centers called PS1 and PS2, or option B PS3 and PSO, or option C PS1 and PS3, or option D none of the above. And the right answer is option A, that is PS1 and PS2. So in the thylakoid membrane, two kinds of chlorophyll centers are present that are called PS1 and PS2. So the last question of this video is, in the initial reaction of the Kelvin cycle, a 5 carbon compound and 1 carbon compound react to form one 4 carbon compound and one 2 carbon compound, that is the option A, or option B, one 5 carbon compound and one 1 carbon compound, or option C, there are two carbon compounds, or option D, two 3 carbon compounds. Here I have a mistake, that is here 3 2 carbon compounds, and option D 2 3 carbon compounds. And the right answer about the Kelvin cycle is option D, that is the product is 2 3 carbon compound. So after reaction with the 5 carbon compound, that is Rubisco, and 1 carbon compound, that is CO2, they produce 3 carbon compound. Okay, that is the phosphoglyceric acid, that is 2 phosphoglyceric acid, that is the 3 carbon compound. Okay, so these are all about this MCQs on biochemistry in this part 6 video. So thank you for watching this video.